Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Pros and Cons of Adaptive Designs, a leisurely conversation between a clinician and two statisticians. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank IDDI, who has helped develop the content for this presentation. International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, and Houston. And now I would like to introduce our first speaker and moderator for today's event. That is Everardo Saad, MD, Senior Oncology Consultant for IDDI and Scientific Director and Chairman for Dendrix. Now, Dendrix is a medical education, communications, and research company based in Sao Paulo, Brazil. He graduated in medicine and trained in internal medicine in Sao Paulo and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. He has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. And our second speaker for today's event is Mark Boyes. He holds a doctorate degree in biostatistics from the Harvard School of Public Health. He is the founder of the International Drug Development Institute and of Clue Points, and an associate professor of biostatistics at Hasselt University in Belgium. He's published extensively on cancer clinical trials. And our third speaker for today's event is Thomas Berzakowski, PhD, VP of Research for IDDI. Thomas Berzakowski has been working as Research Professor of Biostatistics and Bioinformatics at the Center of Statistics of Hasselt University since 2002. He held a visiting professorship position at the Department of Medical Epidemiology and Biostatistics at the Karolinska Institute in Sweden. In 2009, he joined IDDI as Director of Research. Thomas has published methodological papers on topics like surrogate endpoint validation, meta-analysis, missing data, and survival analysis. And now, without further ado, I would like to hand the mic over to Dr. Everardo Saad. You may begin your presentation when ready.
Thank you, Andrew. Hello to all of you, and thanks for attending this webinar. As the clinician, I will have the role of moderator in this webinar, and I will rely on both Tomasz and Mark with their expertise to guide myself and my company in developing a new agent. So throughout the next hour or so, Tomasz will be in favor of adaptive designs, whereas Mark will defend the classical designs. However, we should keep in mind that these opposite stances are not their personal positions, nor IDDI's positions, and we chose to organize the webinar like that just to improve its dynamics. So now let's begin with our story here. If I may have my first slide. So my company has developed a new platinum agent, and we think it could be active in various tumor types, and it appears to be less toxic than other platinum compounds. Now we need to do phase 1 studies in order to determine the recommended dose for further development. We expect that the dose-limiting toxicities will be myelosuppression and peripheral neuropathy. And from preclinical models, it appears that 15 milligrams per meter squared will be the starting dose. And we think we can increase that dose by approximately 50% until we see grade 3 toxicity, and then by approximately 33%. So Mark, what do you think would be an ideal phase 1 trial design?
Okay, good afternoon or good morning, everybody. My view is that I should defend the classical designs, and the classical design for such a trial would be a so-called 3+3 design, also called a modified Fibonacci design sometimes. But the idea of this design is rather simple. We start at a very low dose level, the lowest dose level that is suggested by preclinical experiments. Then we treat 3 patients at these low dose levels. If there are no dose-limiting toxicities, DLTs, then we can increase the dose to the next higher dose level, and we continue to loop and increase the dose until we hit at least 1 DLT. If we hit more than 1 DLT in the first 3 patients at that dose, then the MTD, the maximum tolerated dose, is reached and the trial terminates. If we have 1 DLT in 3 patients, then we have to add another 3 patients. That's why it's called the 3+3 design. And then again, if there are no DLTs in this next cohort of 3 patients, we can increase the dose and loop back again. If there is at least 1 DLT in that next cohort of 3 patients, then again we've reached the maximum tolerated dose. So this is a very simple design that just consists of increasing the dose until at least 2 patients have a dose-limiting toxicity among 3 patients or 6 patients. Now, after that dose-limiting toxicity has been reached and the maximum tolerated dose has been reached, we can do an expansion cohort at a dose that is just under the MTD, and that is called the recommended phase 2 dose for further trials. The advantages of a classical design are many. First, it's simple, it's very safe, it's been shown to work well in many, many instances in the past. It's been very popular and it's still popular today. And because it is so well known, it is very easy to pass a phase 1 trial built on a 3+3 design at an IRB. And so the operational aspects of these designs are well known and therefore would cause no problem or no pushback from the IRBs. The expansion cohort, if we restrict it to specific tumor types, for example, you might be interested to test your new platinum compound in non-small cell lung cancer, is really like an embedded phase 2 trial in the phase 1 trial. And it's used to better characterize the dose and or to see first signs of efficacy at the recommended dose. So I think this design has a lot of advantages, and that's what I would recommend.
Okay, yeah, it sounds well known and safe enough. And in fact, we want to test our agent in non-small cell lung cancer. But then when I look at this graph that shows the number of patients and the dose levels, and considering that the red squares are patients with DLTs, those limiting toxicities, I'm a little concerned that too many patients will be treated at what might be very low doses in terms of treating their tumor. Is there a way around this, Tomasz?
Well, yeah, I mean, exactly. It's one of the problems which I see with the 3+3 design is that many patients are treated at subtherapeutic doses, and you would like to skip these doses, those doses which are too low and which are safe, and to rather quickly go to the doses which are perhaps more therapeutically efficient. One of the problems also with this design, which Mark was presenting, is that it's an ad hoc because it does not guarantee that the MTD, claimed in the design corresponds to a given percentile of the dose response curve. There's no mechanism there which would actually guarantee that you are really targeting the dose which is actually giving you a probability of DLT at a particular level. Now, also the size of expansion cohort is not really justified, and as such, it's also ad hoc chosen. And also, I mean, this situation, for instance, we are we have an agent which we know, so we could have a good guess of the dose response curve, and therefore we could use it in actually selecting the doses which might be used for next patient. So this is actually something which could be used in a more adaptive design, and there is a design like this. It's called continual reassessment method. And essentially, the design starts from as we are in the situation, as you are in the situation, Everardo, that we could have a guess about the probability of DLT at one of several doses. Say we could select 8 doses for your trial, and we could postulate based on the data collected thus far that, for instance, we have a probability of DLT at each of the doses. And we could use this information to assign the doses to the next patients and essentially evaluate the response of the patients and then choose a dose which is more suitable for the next group. So essentially, apart from this prior probabilities, you should propose some kind of a dose relationship model. This may be this logistic model. And let's say that you could start with a model. This model is parameterized with one single parameter, A. And let's say that we would start assuming that parameter A is equal to 1, and then we could essentially choose like the blue curve from all our other curves, and this would be our initial guess, right? And then we could start as follows. We could we could assign the first patient to the dose which, according to this dose response curve, to our prior guess, is closest to the target, let's say 33%, okay? And then the next patient, so we would observe this patient and we would look at the results in terms of the toxicity. And based on these results, we would update the model, okay? And according to the updated model, we could see which dose is closest to the 33% the target probability of DLT, and assign this dose to the next patient. You know, treat the patient, see whether he or she experiences the toxicity, and taking this observation, this result, and the result for the previous patients, update the model, choose the next dose, and so on. So this is like an adaptive scheme, which would essentially mean that after observing a response in terms of the toxicity to a patient treated at the current dose, we update the model, and we then select for the next patient the dose which is the closest to our target dose-limiting toxicity. So in a way, adaptation here is the selection of the doses which are you know, selected in their function of the target level of toxicity. And this design is flexible, and also in terms, for instance, of the stopping criterion. There's no really fixed stopping criterion which you have to use, but there are several which you could use. For instance, you could use as a stopping criterion, you could use a fixed sample size. You could say, okay, I will stop this design when I reach, for instance, 30 patients, okay? Or what we could say is you could decide that you could stop the design when there's a fixed maximum number of the patients treated at a particular dose. You can have as a stopping criterion a fixed precision of the probability of the DLT estimate. You can look at the probability of assigning the same dose level for the next few patients, and if this probability is high, it would mean that you would essentially assign the same dose for the next patient, so you may as well as stop, or other criteria. So essentially, the idea is that you have this adaptive scheme of assigning the doses, which try to target the dose at the level of toxicity which you aim at, and you can stop, you know, depending on one or other criteria which are most suitable for you in terms of the inferential or in terms of the logistical constraints. Now, an example is shown on this slide. So essentially you can see on the x-axis that you have consecutive patients. On the y-axis you have dose level assignment, and you can see that, for instance, in this case the first patient was assigned to the third dose, which according to our prior knowledge was the closest to our target. Then this patient didn't have toxicity, so we went up to dose 4. For the next patient, for this patient, we observed a toxicity, so we might have, the model was updated. It might suggest that we should go lower, perhaps to dose 3. Then we would assign the next patient to dose 3. If they don't observe toxicity after the 6th patient, we would go up, and so on. And what you can see, for instance, is that as of 10th patient, we essentially have a long streak of 11 patients or 10 patients or 9 patients who are actually assigned the same dose, and irrespective of the fact that the patient number 13 experienced toxicity, the next patients are assigned the third dose. So as you can see, this is most likely our MTD. And as you can see, there are many more patients assigned to this dose that as if you would assign them in the 3+3 design, because here you can see that there are 9 patients, or actually, yes, 9 patients assigned to this dose, and in the 3+3 design, the maximum is 6, for instance, right? So you can see that CRM tries to put as many patients as possible around the dose which ultimately may be your MTD, and in this way you gather much more information about this dose or its neighboring doses than in the case of the 3+3 design. So this is actually a very nice feature of the CRM.
Yeah, okay, yeah, I like that. I like that feature of having many patients around the MTD, and also the flexibility of the stopping criteria. But then, Tomasz, don't we run into the opposite problems? If you look at this graph, you see that very few patients, in fact, no patients have been treated at those levels 1 and 2, and we might need to have information on those levels. What do you think?
Yeah, but I mean, this is just essentially the way we started it, right? So what I said is that we start from those 3 because our current guess says that it's closest to our target. But CRM is very flexible. You can actually overrule this prior belief, and you could start at low doses, right? And you can actually populate the low doses if you want, okay? And if you are concerned, for instance, that the model could indicate that you have to increase the dose by 2 dose levels, right, and jump to toxic region, you can actually limit the increases of the dose for next patients only to 1 dose level, for instance, and then to control the probability of so-called skipping doses. You can, in order to, if you want to start at low doses but you want to go quickly through them, you can have schemes which are called accelerated titration, where in order to go through the initial doses quickly, you can actually increase the number of patients assigned to a dose level. Instead of doing this for each patient, you could actually assign 2 or 3 patients to the same dose, and in this way sort of like speed up the acquiring of the information for the model update. You can overrule the model recommendations. If the model, for instance, would indicate, okay, you should go 1 dose up, but for some reason you don't feel that this is appropriate, you can essentially say, no, I will put the next patient still on the same dose just to acquire more knowledge, and there's nothing which prevents you there, because then essentially you gather extra information on the previous dose, then you will update the model and you will evaluate your decision in the function of all previous observations. So it's a very versatile and very flexible design. So I know that Mark likes 3+3, but I would sort of try to argue in favor of CRM for you, Everardo.
All right, I'm convinced. So now let's consider that we run our phase 1 trials using the CRM, and now our agent is ready for phase 2. And let's consider that the dose determined in phase 1 is not questionable. It's a correct dose. So now we'd like to know if this agent is sufficiently active to move into phase 3. And we think a single agent response rate of 15% in second-line non-small cell lung cancer would be of interest. So going back to you, Mark, what phase 2 trial design would you recommend at this point?
And again, I would take the view that a classical design might well do the job, and we have been using 2-stage phase 2 designs for a long time. Rich Simon proposed a 2-stage phase 2 design back in 1990, and this has been extremely popular. There is also a version of the same design with 1 stage only proposed by Fleming, but essentially this is the same idea. You just try to test if the response rate that you will obtain with your new drug is in fact within a certain range, and what you don't want is that let's suppose the response rate is less than 5%. That would be completely unacceptable for this new drug. And you hope that the new drug will give it a response rate of at least 20%. So the hypothesis you test is that the response rate is less than 5% versus more than 20%. And you can pre-specify the error rates, alpha and beta, against these two thresholds of activity, 5% being the low level and 20% being the high level of activity that you target for this particular drug. So if we were to test these hypotheses, less than 5% versus more than 20% in a single-arm design, so it's not a comparative design, it's just a one-arm design where we test the response rate of the drug against specific thresholds, and with the error rates indicated here, 5% for alpha, 10% for beta. The design would consist very simply of treating a cohort of patients, a first cohort of 20 patients. If we have no response, we can stop and claim that the drug is not efficacious. If we have at least one response, then we add a second cohort and we evaluate response in the second cohort of 20 patients. In the end of the trial, at the end, if we have less than 4 responses in total, we would claim that the drug is insufficiently active. And if we have more than 5 responses or more, we would conclude that the drug has evidence of efficacy, and then we move the drug forward to phase 3. So this is, I think, a very solid and well-established design. The operating characteristics of this design are well known. They can be easily studied. But the problem in this design, as in many other designs, is that the response rate, and as well as other efficacy outcomes, is very often driven by patient characteristics as well as, if not more than, by the treatment activity. And so randomization is often required to check if the response rate or the outcome of interest is consistent with historical data. So I think today people increasingly want to do randomized phase 2 designs where they randomize patients between the new treatment, in your case this new platinum compound, and some standard treatment that would be appropriate in the indication, perhaps a well-known platinum compound that you could compare the new compound with. And there are 2 main approaches to doing this randomized design. The first approach is, in fact, as I just explained, to test the response rate of the new compound against pre-specified thresholds. And in that case, the standard treatment arm, which is a randomized control, just serves as an informal comparison, an informal reference, just to see if you are within the range that you expect as far as response rate in this particular patient population. And the other approach is to actually test for a difference between the response rate on the new treatment and the response rate on the standard treatment. In other words, Rn minus Rs. And then you test that difference, but using relaxed error rates, alpha and beta. If you didn't use relaxed error rates, you would actually be doing a randomized phase 3 trial, which we will discuss later. So this randomized phase 2 design could well use a Simon 2-stage or a Fleming 1-stage design, and that would be the classical approach of testing this new drug.
Okay, yeah, we like the randomization regardless of the approach we take. But then, Mark, I have a question for you. Since our agent is new and the control group will be an established drug, is there a way we can have more patients getting our drug so we can have more information, whereas we don't need so much information for the well-known standard treatment? Can we do that?
Again, there is a simple classical way of doing this, and it's to randomize more patients to the new treatment than to the standard. There's really nothing against randomizing twice as many patients to receive the new treatment as compared with the standard treatment, or even 3 times more patients receiving the new treatment as compared with the standard treatment. The allocation ratio is really your choice. And in fact, an unbalanced design will give you much more information on the new drug, which is where you need to have more information, and less information on the standard treatment, which you already know about. So it makes sense to have this imbalanced allocation ratio in a classical sort of design.
Yeah, well, that's a good idea actually, but you could push it even further. So you could use adaptive randomization. The idea is to allocating, indeed, the same idea is to allocate more patients, but in this case to allocate more patients to the treatment with the better outcome. So essentially what you could broaden this to is to have many treatments and to try to try to allocate the treatments adaptively to those which seem to be working. So this is the idea of so-called the battle trial, where the endpoint was the progression free at 8 weeks, and then a Bayesian approach was used to construct prior distributions for the endpoint in multiple biomarker times treatment subsets. Okay, and then the idea was to assign more patients to more promising subsets, and the idea was that subsets that have insufficient activity would be stopped early for futility. So essentially, what you have in the battle trial is that you have several groups, biomarker groups, based on molecular biomarkers, and essentially you have also a group of treatments. So the idea is that you could use adaptive randomization to so all the patients go through this through the biomarker profiling. Then we have the prior distribution construction, and then adaptive randomization is used to allocate treatments to the appropriate subsets of the patients based on the observed outcomes, so observed endpoints for the patients treated so far. And in this way, the idea is that you assign actually only those treatments which seem to be only to those biomarker subgroups which are actually benefiting from them. So you are not constrained by any ratio, a priori setup ratio of randomization of one treatment to another, nor you are in a way constrained to select nor in a way you are constrained by the number of the subgroups, for instance, because you can drop subgroups and just continue with those subgroups which seem to benefit from some treatment. So you can push this idea of putting more patients on some treatments for further than just using constant and fixed allocation ratios.
Now this is, of course, it's a fantastically innovative design, but is it not too good to be true? I think there are many issues with trials like this. First of all, what exactly are the hypotheses that you test? And can you really investigate as many as 4 treatments and 5 biomarkers groups, which is 20 subsets, 20 combinations, if you like, of biomarker by treatment in a modest sample size? I mean, are you going to need, several thousands of patients to do this reliably? We have no idea what the error rates are. So I know that I'm talking about this design as a conventional statistician, but what are the error rates? Do you have any control over the error rates? And by error rates, I mean the error of a false positive and the error of a false negative. Could you miss some treatments that would have worked, or could you actually falsely identify treatments that you think work and in fact don't work so well? The endpoint in your trial is PFS at 8 weeks. That is an endpoint that is equivalent to a response rate, so it's an endpoint that has advantages but is also quite limited. It's a binomial outcome, it's a binary outcome, and perhaps you should use some biomarker-driven endpoint to have more power to correctly select the treatment by biomarker combinations that you think work well. And then, of course, you didn't say anything about the priors here. You said that you would use a Bayesian design, but I haven't seen the priors that you assumed for each of the biomarker by treatment combination. And then finally, Thomas, my biggest concern about this battle design is this adaptive randomization. Is it statistically or ethically desirable to have an adaptive randomization? I personally don't think so, and I'm not the only statistician to think that. There is a paper by Korn and Friedlin in the Journal of Clinical Oncology that I would point you to, which I think gives an excellent overview of the problem. And they conclude by saying that they prefer a fixed, let's say 2-to-1 or 3-to-1 randomization ratio rather than this adaptive randomization. So I'm not convinced that you need the complexity of the adaptive randomization for a trial like this.
Yeah, okay. I think for phase 2 we'll have to spend some more time within the company thinking what way we should take. But let's consider we run a phase 2 trial and found a response rate of 20%. And we think this is very encouraging because the historical response rate is 10%. So now let's move into phase 3, and we think that a response rate of at least 15% would be very, very good. So coming back to you, Mark, what do you think should be the phase 3 trial design?
And as you expect, I would recommend the classical phase 3 design. And again, I'm summarizing here what you just told me. You're looking at the difference in response rate, which in fact in practice we would very seldom do. But let's suppose, just for the sake of this discussion, that response rate is the outcome of interest. In general, it would more be like PFS or OS. But let's suppose we are interested in this difference in response rates. What you said is that you had a 10%, you expected a 10% response rate on the standard treatment. Maybe you hope for a 10% improvement of that, but you would already be happy with a 5% improvement over the 10%. And let's suppose we use conventional levels of significance and power, 5% for alpha, 20% for beta, and that would require a sample size of about 400 patients. Now, I think you can look at two extreme assumptions here. You can assume that you want to look at the smallest possible effect that is of interest. Let's say it's 5%. You said that a 5% improvement in the response rate would make you happy. So that means that you might actually want to power your trial for as little as a 5% improvement. And you would be cautious to do that because early trials, as we know, very frequently overestimate the true outcomes of interest. And that's why so many phase 3 trials, it's because the phase 3 trials are designed based on phase 2 results. And more often than not, phase 3 results are not as good as phase 2 results. So I think it's very cautious to look at the smallest difference possible that would be of interest. Now, of course, you could also take the other view that to say, well, I'm only interested in this drug gives me a really, really big improvement, perhaps something like a 20% improvement. And that, of course, would lead to a much smaller trial. So if you did a trial using the smallest effect of interest, you would need 1,400 patients. If you did a trial using the sort of much larger, more interesting effect, you would need only 120 patients to look at that situation. So you have to make a choice between these two extremes. And perhaps it's fair to say that big pharma tends to take the view that small differences are worthwhile and a drug might well work and be, useful on the market, even if its effect is rather small. And that's why they tend to do large phase 3 trials. Simply because, they hope that the drug might be more effective than the smallest effect that's clinically worthwhile, but they sort of plan the trial in the worst case scenario. So they would plan a trial for 1,400 patients and hope that an interim analysis might stop earlier if the drug has more efficacy than expected. Whereas it's fair to say again that small biotechs, being more limited in their budgets, have to settle very often for smaller trials. They'd be looking at a major effect of their new drug and hope that they will see an effect in the small sample size and be able to adaptively increase the sample size to reach a larger sample size and have a convincing result. So I'm going to just show here a classical approach to this problem, which is to do a group sequential design, assuming that the difference of interest is the smallest difference that we said was of clinical interest, 5%, plan the trial with the parameters pre-specified, alpha 5%, a power of 80%. We need 1,400 patients maximum, but then we do 3 interim analyses and a final analysis in a group sequential fashion. So we analyze the data every time we have a quarter of the patients randomized. After 350 patients, we can look at the data already, and if we have a difference in favor of the new drug of 15% or more, then that would produce a p-value for the test, comparing the new treatment with the standard treatment, a p-value of less than 0.001, and that would be sufficient to stop the trial already, claiming that we have a significant difference. If that interim analysis didn't stop the trial, then we would go to the next one and repeat this, 3 times. The next analysis would stop if the difference was 7% or more. And finally, the third interim analysis would stop if the difference was exactly what we anticipate, which is 5%. Of course, we hope for better than 5%, but if the difference was, as we think, 5%, we would stop after 1,000 patients or so. And then if we didn't stop there, we would go to the final analysis, and we would then be able to detect a 4% difference. Now, the trial, as I said, would stop at the third interim analysis if the difference was what we expect it to be. We could also add to this design a futility stopping boundary, which is exactly the same as an efficacy stopping boundary, but except it's the exact opposite. We stop the trial if we have no evidence of efficacy of the new drug compared with the standard. So if at the first interim analysis, for example, we saw a difference against the new drug of 3% or more, we would stop right there and we would say it's futile to continue this trial because we have evidence that the drug doesn't work. And this, by the way, I didn't say, is using an O'Brien-Fleming stopping boundary. You could use other boundaries if you preferred. You have a lot of choices. You have a lot of flexibility in terms of the kinds of boundaries you want to use. You could even use a different boundary for efficacy and for futility. You might be more aggressive to stop for futility than you are to stop for efficacy. So this is a very, very flexible type of approach which allows you to look at the data multiple times and stop the trial the minute you have enough evidence that the drug does work or does not work. And so I would argue that with the group sequential design, you have the instrument you need to test your drug in a very flexible manner, and I would argue that this is probably in most cases what you want.
Well, yeah, I mean, that would be a good argument, but my question to Everardo is how much money does he have for his design, for his trial, actually?
Not much.
Not much. Okay. Well, that's what I expected. So, well, then actually, it might be worthwhile to do an adaptive design starting from the other end, as already Mark alluded to. So, you could actually start more or less with the same assumptions, but start with a difference of interest of 20%, right, which might be large. But this means that it would allow you to use a sample size of about only 200 patients, right? And then you could do two analyses after 60 and 120 patients, right? And you could, if you have the treatment which really works, you could stop if the p-value is less than 3,000 at the first interim, or about 5% at the second interim, which boils down to stopping when the minimum difference is 30% at the first interim or 14% at the second interim, right? Now, the thing is that what you could do is essentially, okay, if the treatment, if you do look at the interim results, you could actually increase your sample size adaptively if you realize at the interim analysis that the conditional power, so the power of rejecting the null hypothesis given the data, given the difference you have observed thus far, is too low. For instance, if it's low, it's below 80%, which you pre-planned, right? The idea would be that if you see at your interim analysis that your treatment doesn't do as good as you think, you could actually try to increase your sample size, and only then, of course, you would have to look for the extra money, but only then. So the idea is that you could have a flow like here. At the interim, you calculate the conditional power, and you ask the question, is it high enough? For instance, is it 80% or more to reject the null hypothesis at the end? If yes, just continue your trial, okay? But if no, you can consider your sample size increase, all right? Now, you can put yourself a limit. So, for instance, you don't want to spend, you don't want to enroll 800 patients. So let's say that if you plan 200 patients and if you have to double this number, you can see whether your increased sample size is below this limit. If yes, you increase your sample size and you continue. If no, if your sample size is much larger that you can that you can ever consider, well, then you just stop trial for the futility, and you will not invest you will not waste your time and money on something which does not bring you the answer you were hoping for. So it's just this idea of computing the conditional power and based on the results which you observe now, and continuing or not depending, or increasing your sample size or not, depending on this conditional power. So let's look at an example. Let's say that you might be in a situation after 60 patients where you have 3 responses in the standard group and 6 responses in the new group, so in the new treatment. So if you look at the conditional power, so the difference of the observed effect is 10%. So the conditional power is equal to 27% given this observed effect. Is clearly too low. So you might want to throw extra patients to reach back again to 80%, to increase your chance of rejecting the null hypothesis at the end of the trial. Okay, but now you know that your that your difference is 10% only. It's far away from your 20% which you hold. So in order to do this, you would have to increase your sample size to 360 patients, right? So just below the 400 which you might afford. So a little bit less than doubling. Okay. And this means that you could do this in such a way that you still keep the planned alpha type 1 error probability, and you go back to this conditional power of 80%, so increase your chances that at the end of the day your trial will have a chance to reject the null hypothesis, and you will have a chance to claim that your treatment is effective. So this is like, you know, starting low and modest, perhaps with the hope for a good result, and, you know, increase along the way if you need it to increase your chances. But if your treatment is very good, you will perhaps not have to increase anything. But if you will have to increase, you can. There is a design like that, and this would allow you to do the trial, perhaps to start the trial from the other end when the limited resources are available and when you need to take care into the fact that the number of the patients which you may put on the trial is limited. How about that?
Yeah, okay, Tomasz. Yeah, I like that. But are there risks to doing that, Mark?
I think there are risks. I mean, this is, again, a little too good to be true because there are many operational difficulties with doing adaptive designs. First of all, Tomasz didn't mention this, but the interim analysis upon which the decision would be made to increase the sample size has to remain blinded. You as a sponsor will not be allowed to see that analysis, because if you were to see that analysis, that might create a bias for the rest of the trial. So you would have to have this analysis done and evaluated by an IDMC, an Independent Data Monitoring Committee, which would then take the decision or give you the recommendation to increase your sample size or not. The other operational difficulty which we often face in cancer is that you have to have endpoints that are observed fast and reliably. If we're talking about an overall survival trial, you know, by the time you have enough events to adapt your design, you will have accrued all the patients, and it will be very difficult to reopen accrual after observing more events. So I think the timing of these interim analyses is a difficulty that, of course, we didn't have in this particular case because we looked at response rate, which is seen immediately. But if we looked at any time to event endpoint, we might have that difficulty. The agencies, the FDA and the EMA, may have some concerns, indeed, if the interim results are known, even indirectly, because if you increase the sample size, many investigators will feel that the effect of the treatment is in fact less than you were hoping, and therefore they may then begin to enter different types of patients in the trial because they now know that the effect is not as good as you perhaps were hoping, and therefore they may enter maybe better patients or worse patients. We don't really know how they will react, but they may have a reaction, you know, to this news, indirect news, that the sample size has been increased because the efficacy was not as great as you were hoping. And so, in fact, you have these operational difficulties, and plus, something Tomasz didn't mention is that if you compare the efficiency of an adaptive design to a group sequential design, it's always less. So an adaptive design is in fact less efficient statistically than a group sequential design. There is a good paper on this by Butch Tsiatis and Cyrus Mehta, where they show that for any adaptive design that you could think of, there is always a group sequential design that outperforms it in terms of efficiency. And so the question then is, if you know that you don't have an optimal design in terms of efficiency, why would you want to use it?
Well, yeah, but yeah, it's a good point. But I mean, group sequential designs are really aimed at, you know, early stopping, right? But you know, adaptive designs can bring you much more flexibility. As we saw, we could increase or decrease sample size, but you could also think about, you know, stopping a treatment arm, for instance, if you have multi-arm trial. So there is actually broader flexibility in adaptive designs. Essentially, you can sort of like make them robust against the assumptions which you made at the design stage related to your, for instance, perceived let's say, background rate of response in the control group, which would influence the number of the responses you observe in total and therefore the power. Actually, regulatory agencies encourage pre-specified flexibility, so we could say we could actually target the design towards the particular aspects which we want to be flexible about, especially if it speeds up the development and or increases the chance of success. So we could, it is actually, these type of designs are encouraged as long as, of course, we can properly use them. Obviously, I mean, it's easier and less expensive for biotechs to start small and increase sample size, so it's actually your situation, Everard, I guess. And essentially, you know, it's better to adapt than to fail. So, I mean, if you, as in the example I just showed, if you see at the interim that actually your treatment you know, is not performing that well as you hoped for, but it's still showing a reasonable, interesting effect. It's like doubling the response from 10 to 20%. I mean, then if you adapt, you can still have a chance to actually succeed. Another idea which you can have is something which is quite interesting, and especially nowadays when biomarkers become available, it can be used. It's adaptive enrichment. So let us look at this design where, again, we have a response rate on standard treatment of 10%. We want to see whether the response rate on new agent can be increased to 20%, significance level 5%, 400 patients. If we just use all the, if we just calculate the power, it's 80%, you know, on all. Now, just imagine that this response rate on new agent is slightly lower than you hoped for, just 17%. What happens is that all of a sudden your power drops to 53%. It's almost like tossing a fair coin. So this is really like a small difference in terms of the response rate, which can hugely impact the probability of finding the difference, claiming the difference, and therefore the success of the trial. Now imagine that you could split your population into two subpopulations based on some biomarker. Okay, there are molecular biomarkers in oncology like EGFR mutation or ALK rearrangement or other type of mutations. So you could for instance imagine that this 17% response rate on the new agent can be seen as a weighted average of a rather high response rate, 30% in a biomarker positive group, which constitutes about 30% of your population, and rather disappointing response rate of 11% in a biomarker negative subgroup, which constitutes 70% of your population. Okay, so now what you see is that the 17% is due to this weighted average. So what you could do is you could actually at your interim decide to go on only for the biomarker positive patients. And if you do this, and so if you assume that the second half of the trial will only include the biomarker positive patients, and if you design the trial in an appropriate way so that the overall type 1 error probability is controlled at the alpha level, then your power of the adaptively enriched trial can jump to 95%. So 53% if you consider the whole population, but 95% if you adaptively enrich your trial in the middle by allowing enrollment of the patients in the more, let's say, suitable subgroup of the patients. So this is a very interesting idea, again, with the adaptive approach in mind. So this can be shown in a sort of like a flow chart when you can again combine this with the increase of the sample size, for instance. So you calculate at the interim analysis, you can calculate your conditional power for the whole population, okay? For the whole target population. If this conditional power is large, then 80%, okay, you continue your trial unchanged, all right? However, if your power is less than 80%, as before we could say, okay, we could increase the sample size. And we could ask, is this increased sample size less than the limit which we pose ourselves, like less than 400 that you would allow yourself to go for a maximum? Now, if it is still within these limits, you can increase the sample size, so we have the previous idea. At the interim, you adaptively increase your sample size and you continue. However, if you cannot afford that many patients, you could consider enrichment. So what you could consider is you could say, hey, hold on, let me look at this subpopulation where I had a higher treatment effect, right? And let me see if I could enroll only this subpopulation in my trial in the second half of the trial. So what you could do is you could take a conditional power calculation for this subpopulation, and again see how much you would have to increase your sample size if you would limit the second half of your trial to these patients. Now, if this increased sample size is less than the limit which you pose for yourself, like this 400 for instance, you can increase the sample size and go for this subpopulation only. And as I showed you on the previous slide, you could actually effectively increase the power of your trial with including only these patients into your trial. Of course, if also in this subpopulation, the increase of the sample size would go over your limit, well, there's nothing else what you can do. You would just stop the trial and you would claim futility. But as you could see, you could also use an adaptive design in the sense that you could go on and try to zoom in on the subpopulation which may be really benefiting from that, may be showing signs of benefiting from the treatment, and therefore you could increase the chances of showing the effect of the treatment, at least in this subpopulation. So the adaptive design allows you to do this, and well, this is not something group sequential designs can swallow. So Mark, of course, is right when pointing to these problems with the adaptive designs in comparison to group sequential designs, but group sequential designs will not allow you for that flexibility that adaptive designs may offer you. So I would think that this is an viable alternative for the phase 3 design for you, Everardo.
Okay, thank you both Mark and Tomasz for these very insightful and I should say well-timed discussions. For those of you attendees who joined a bit later, I'd just like to remind you that these opposite stances taken by Mark and Tomasz regarding being in favor or against adaptive designs were just used for the sake of illustration. These are not necessarily their own personal views. So I believe it's now fair to say that the take-home messages I will take from this webinar are these. Adaptive designs can be helpful throughout drug development, especially in early phase trials where the uncertainties are higher and the risks lower. It's clear that adaptive designs could be used in phase 1 trials. Perhaps they should be used more. For example, the continual reassessment method. Unfortunately, they aren't used very much in phase 2 trials, but it's unclear the best way to go. There are many opportunities, as we discussed. And in phase 3 trials, the use of group sequential designs with interim analyses appears better than adaptive designs, except when enrichment is possible. That should be considered. And another point is that adaptive randomization is not generally recommended. So I see there are many questions from the attendees, and we'll take some of them, and I will pass the mic to Andrew now.
Great. Well, thank you all for that great presentation. A lot of insight held there, I think, for all. At this point, I'd like to invite our audience to continue sending their questions and comments in right now using that questions chat window for the Q&A portion of this webinar. And like you said, I received a lot of questions here already. Our first question, can the primary endpoint be changed in an adaptive design? Your thoughts on that, please.
Mark, do you want to comment?
Anyone, really.
Yeah, okay. Let me take the view that, yes, theoretically you can change the primary endpoint of a trial midway into the trial. Statistically, there is little problem in doing this. There are methods that allow you to do this and still control the type 1 error of the whole trial. But I think it would be very dangerous to change the primary endpoint of a trial midway into the trial, because then what you do is essentially just test a hypothesis and control the type 1 error for this hypothesis. But the hypothesis is now the combination of two different sub-trials, each of which uses a different endpoint. And I'm not sure that a clinician or a regulatory agency would be able to interpret the data that you would present to them, because the whole trial would be a combination of two sub-trials, each of which would have their own primary endpoint. So I think it would lead to a lot of controversies and difficulties in interpretation. And so even though the statistical aspects of this have been studied, I think it would be ill-advised to change the primary endpoint midway into a trial unless there was a really, really good reason to do so.
Well, yeah, I mean that's also that it would also have to be combined in with power considerations, right? Because in a way, if you change the endpoint, then you would be using it in a half of the trial. Then the question is how much power you may gain and stuff like this. So it is a complex adaptation, and indeed it would be cumbersome.
Okay, well, thank you both for that. Moving on, our next question here. And I believe this was referring to your slides early on in the presentation titled An Example of a CRM-Based Trial. The audience question, what about using a target interval instead of a target threshold and then using distribution instead of a point estimate? Your thoughts on that?
Well, maybe I will take this one. I'm not exactly sure what the question is, but I can perhaps refer to a design which has been recently published in Journal of Clinical Oncology, which is called Modified Toxicity Probability Interval, where essentially what you look at is you look at like intervals in the sense of you look at the classification of doses like which are less than MTD, more than MTD, and like MTD with some interval around the target toxicity level, and then all doses which fall into this range around the target DLT probability are seen as close to the MTD, so you can choose one of them for your for your next phase 2 step, for instance. So there is a design like that which sort of like tries to combine the idea of pre-specified simple 3+3 design with kind of aiming at the target level of the dose-limiting toxicity. So it might be the design which sort of like goes into the direction of this question.
Okay, thank you for that. Our next question. With multiple opportunities to stop the design, we're now in the realm of multiple testing. Do you correct the p-values to take that into account?
Well, let me answer this one from the viewpoint of the group sequential designs. The answer is definitely yes. The group sequential designs specifically calculate boundaries in such a way that the p-values that you would use for the interim analyses and for the final analyses are controlled in such a way that the type 1 overall error rate for the whole trial is controlled. So certainly for group sequential designs, the control of type 1 is guaranteed through the design. And I don't want to preempt Thomas's view, but certainly for adaptive design you would do something similar.
Yeah, I mean there are rules which you can use to combine the p-values from the different interim analysis and adaptive design so that the overall level is controlled. So you also take this, take the sort of like this aspect of inference, statistical inference into account. So you do control type 1 error by appropriate combination of using the p-values and appropriate selection of the levels for the different interim analysis. So in both worlds, if you talk about the control of type 1 error rates, you also you take this into account.
Okay. Well, thank you both for those viewpoints. Our next question, what do you think about continuous monitoring for phase 2 design instead of the classical Simon design?
Continuous,
okay,
yeah, go ahead, Mark.
Let me give it a try.
Yeah.
Continuous, yes, theoretically it's not terribly difficult to have a continuous monitoring. You have sequential design methods. John Whitehead has a very nice book on sequential methods in clinical research. It turns out that in practice, sequential designs are very seldom used, and group sequential designs are much more often used simply for operational reasons, because it's actually quite difficult to do a trial and to update your analysis every time you have a new patient in the study. It makes much more sense to look at groups of patients, simply for operational reasons. And so group sequential designs have become very popular. Now you can increase the number of interim analyses if you want and have smaller groups, but I don't think it's realistic in most cases to have an analysis after every patient. But you could do it theoretically if you wanted.
Okay, thank you for that. Our next question here. Why is the operational bias due to knowing the interim result in adaptive design different from the same situation in group sequential design?
Oh, it is very different. I will let Thomas expand on it, but in a group sequential design, you do not change anything to the design itself. You just look at the data and decide to stop or to continue the trial based on pre-specified interim analyses. But other than these pre-specified looks, you don't change the design in any way. Whereas in an adaptive design, you take actions based on the interim analysis. You decide to increase your sample size, or you decide to enrich the trial, as Thomas explained. So that is a very different type of decision than just stopping or continuing.
Yes, and that's of course the source of the concern. And you already, Mark, alluded to the fact that, you know, it's like the decision to enroll, for instance, other subpopulation of the patients may be conscious, but maybe also driven by the fact that you have some adaptation done along the way, and this may be actually influencing your results at the end of the trial. So indeed, I mean, the nature, I mean, of the adaptations are different in the group sequential and, well, in general adaptive designs, and this is where the source of the problem lies.
And one of the things that the agencies will often want to see is the results of the trial before and after the adaptation. So if by chance your trial outcomes change, maybe because the population of patients has changed, or maybe just because of the play of chance, if you have an outcome which is very different after the adaptation as compared to before the adaptation, you may run into some tricky issues, because then you have to sort of explain why that is. If you've enriched the trial, fine. You can explain that you have better outcomes because, you know, you've enriched the trial in responders. But if it's just a play of chance and you have, let's say, worse results or some, you know, different types of results before and after the adaptation, it opens up a whole range of problems that you wouldn't do if you did a group sequential design.
Okay, thank you. I think we'll push a little bit over time since we have so many questions here to ask maybe one or two more questions. The next question is referring to your presentation on slide 39. And this audience member says, unfortunately, most people will interpret the conditional power as the probability of statistical significance at the end of the trial. I would at least emphasize that these are different and that predictive power is actually the result that gives what most people think comes from conditional power. Your thoughts on that?
Well, yeah, I think that in the question there's already an answer. Indeed, it's that it's not that you sort of like calculate the chance of having significant p-value at the end, right? It's the concept of the, you know, like what is the probability of rejection of the null hypothesis given what I have observed now, right? So it's more or less like, okay, it's if you do the unconditional power, and then you have your 80%, which means if I have this, if I assume that this is my true treatment effect, I would expect that in 80 cases out of 100, you know, at the end of the trial, I would reject the null hypothesis, right? So similarly, you can think about this conditional power calculations in these terms, but you take into account the fact that at this current moment you observed the difference of 10%, for instance, right? So you modify this power calculation, but nevertheless it's not the probability of significant result at the end of the trial, as it is not for unconditional power. I think this is a correct statement, Mark, right?
Yeah, I'm not sure I understand the question, but I think what it points to is that you can calculate the conditional power under different assumptions about the true value of the parameter. Which is, I think, what you also said. So if you now have evidence that the true value of the parameter might be different from what you assumed initially, you can now calculate the conditional power for the initially assumed value, or for the newly estimated value, or for any other value of the parameter of interest. And in fact, if you have a Bayesian type of approach, you can even integrate your conditional power over all the values, you know, using a prior, and then you get the predictive power, which was the word that the person who asked the question mentioned. And so you could indeed do the predictive power calculation as well if you wanted, if you had like, you know, a good prior for the distribution of your parameter of interest. But that is, again, a kind of a complexity of doing these conditional power calculations. You can do it in many different ways, and it's not completely trivial which of the values of the parameter makes the most sense to use.
Well, but that's no different from what you are facing when you start the trial, right?
Absolutely,
I mean,
I agree.
Yeah.
I agree. It's the same problem when you start the trial, except that now you have some data
Exactly.
that you can use to estimate the parameter, even if it's a very imprecise estimation.
Okay, thank you for that. Perhaps one or two more questions at this point. For adaptive enrichment design, then can we use the same sample size calculation than for classical two-arm design?
Okay, I'm not sure. Now, when I heard this, I'm not sure whether I get it in the sense. So probably the question is if we have this. I mean, the power calculations. Sorry, it goes about. Can you read the questions once again, Andrew?
Absolutely. For adaptive enrichment design, can we use the same sample size calculation than for classical two-arm design?
Well, I mean, the adaptive enrichment is again related to this concept of conditional power at one point, right? So you need to sort of like link the calculation of the sample size to the calculation of your conditional power. So it's a little, you know, it's not like the classical two-arm sample size formula, but it's more like, you know, looking at this conditional power formula and then trying to see for the particular sample size which level it reaches. I'm not sure whether this answers the question, but I guess this was my best shot at it to explain it.
Okay, well, thank you for that. I think one last question here. What does the panel feel about minimization?
I'll let Mark to handle this question.
We would need a completely different webinar, I think, to go over minimization, which is really not something that we covered at all today. In short, I think it's fair to say that we love minimization as being the best method in all cases to allocate treatments to patients in randomized trials. Unfortunately, the agencies tend to take a slightly different view, but I have yet to understand why they do so. So that would be my view of it, and perhaps we would need to have another webinar on that topic.
Okay.
And this was a personal view of Mark, actually.
Okay. Well, thank you for that. Yeah, maybe a future webinar. Thank you all for those answers. We had a great Q&A session today. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can direct them to Dominique Grissard at dominique.grisard@idddi.com. And you can also ask her for a copy of the PowerPoint presentation through that email address. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event, and a survey window will be popping up on your screen, and your participation is appreciated as it'll help us improve our further webinars. Now, please join us in thanking our speakers, Dr. Everard Osad, Dr. Mark Boys, and Dr. Thomas Berzikowski. We hope that you found this conference informative today. Have a great day, everyone.
Thank you. Bye-bye.